Hello student, myself Darshna Maru, Matrushi Ramubati and Matrushi Santaben Vidya Science College connected to Sardar Patel Educational Institute. I welcome to all of you on online tutorial. Today we discuss of Sem-5 B.Sc topic named Enzyme Linked Immunosorbent Assay. We all know as ELISA. ELISA use technique for detection of antigen or antibody in 1971 by Pepar-505 and Igm H at Stockholm University, Sweden. Principle of ELISA. Principle is based on the formation of antigen-antibody complex which is detected by detection using enzyme conjugated. The conjugated enzyme act on specific substrate called chromogenic substrate and generate a color reaction product. This product is qualitative or quantitative read using an ELISA plate reader. Here you can see the ELISA reader picture. ELISA kits are commercially available which can conveniently use for laboratory purpose. Here you can see the picture of ELISA kit with Microtiter plate, buffer solution, Microtiter plate having 96 well, so you can easily read loaded 96 sample with their controls. Types of ELISA. ELISA have mainly 4 types. First, direct ELISA, second, indirect ELISA, third, sandwich ELISA, and fourth, competitive ELISA. First, direct ELISA. It is used in the detection of antigen in the given biological sample. Microtiter valves are initially weighted with antigen to be detected, which is followed by an antibody linked to an enzyme conjugate. This follows the addition of substrate which produce color detection using ELISA detector. Here you can see the figure of direct ELISA. Well coated with antigen, it will bind with primary antibody and enzyme link substrate from color combination and it will detect by ELISA reader. Second type, indirect ELISA. It is used for detection of an antibody in the given sample. Microtiter valves are initially coated with antigen specific for antibodies to be detected, followed by the addition of sample. Enzyme conjugated secondary antibodies is added, followed by the substrate which form a color reaction product. Here see the picture of indirect ELISA. Well is coated with antigen. It will bind with primary antibodies. It will again bind with enzyme linked secondary antibodies conjugate. Then added substrate due to enzyme activity colored complex form and it will be measured by ELISA reader or detector. Third type is sandwich ELISA. It is used for detecting antigen in the given sample. Microtiter valves are initially coated with monoclonal antibodies. Rays against antigen to be detected followed by addition of sample. Any trace of antigen is detected by adding primary antibodies, then enzyme conjugated secondary antibodies and the chromogenic substrate added. See the figure of sandwich ELISA. Antibodies is attached with well, then bind with target antigen and serum and bind and form antigen antibody complex. Then again bind with enzyme link secondary antibodies conjugate. This will bind with substrate form color complex. This stage form structure like sandwich. So you can say the sandwich ELISA. Fourth and last type of ELISA is competitive ELISA. This variation of ELISA is used to quantitative estimation the amount of antigen is the given sample Antigen and antibody are initially incubated so that the antigen antibody complex, this mixture added into microtiter well coated with synthetic analog of antigen to be detected. Any free antibodies bind with antigen with coated bottom of the well. Complex will estimated by enzyme conjugate secondary antibodies for chromogen detection. More amount of antigen in sample and lesser is the antibody available to bind microtiter well. Here you can see the figure of competitive ELISA. You can see the sample which have excessive amount of antigen, it will bind with antibody for antigen antibody complex. Still, some antigen we unbind, this will bind with conjugate and lesser amount of color complex form. Application of ELISA. ELISA can detect both antigen and antibodies. It is useful tool for determination serum antibody concentration. It has also found application in the food industry in detection potential food allergens such as milk, peanut, walnut, almonds and egg. Detection of Mycobacterium antibody in tuberculosis. Used for detection of Hepatitis B marker in serum. Use detection of enterotoxin of E. coli in fishes. Detection of HIV antibodies in blood sample. Detection of syphilis, IgG or IgM, H. pylori, IgG and antigen detection. Human allergen specific IgE and IgA ELISA. Advantages of ELISA. Sensitive assay equipments are widely available, no radiation hazardous, reagents are cheap with long shelf life, qualitative and quantitative procedure. ELISA can be used on most type of biological samples such as plasma, serum, urine and cell extract. Disadvantages of ELISA. Only monoclonal antibodies can be used as match pair. Monoclonal antibody can cause more than polyclonal antibody. Negative control may indicate positive result if blocking solution is ineffective. Enzyme or substrate reaction is short term, hence color must be read as soon as possible. Now the conclusion of ELISA. Enzyme linked immunosorbent assay is a novel technique useful in detection and antigen and antibody present in the given biological sample. Besides its disadvantages, the technique is being widely used in diagnostic and drug screening. Chromogenic detection method used in ELISA is convenient and sensitive for any assay and it's hazard free. Thank you for watching.